Good morning and welcome to BPI TV. I'm Ann Montgomery, Editor-in-Chief of Bioprocess International, and I'm here today with Sigma Mostafa, who is Vice President of Process Development at KBI Biopharma. Good morning.
Morning.
Would you like to begin by talking about KBI's approach to speeding the process toward the IND?
Absolutely. So, KBI has been focused on being one of the fastest to IND in the market because that's one of our clients' definite ask. However, when we do that, our thought process, our philosophy has always been that we want to achieve it through our technical depth and the technologies that we employ, right? So that by going to IND fast, we are not compromising on the quality of the work or the robustness of the process that goes to IND, right? So essentially, the three things we look for in getting fast to IND is a robust cell line, as well as integrated approach across functional areas, so upstream, downstream analytical formulation development, as well as being able to essentially integrate the high throughput technologies. And all of those three aspects have helped us to be one of the fastest in industry. And now that Selexis is part of the same umbrella under JSR, our parent company, we have been able to really integrate very well because of the strong cell line they bring into the equation.
Okay. How are you approaching rapid late phase development and reducing the timeline for process characterization?
Sure. Yeah. So what we find nowadays is clients are not only asking for going fast to IND, but they're in fact saying going fast to BLA. So of course, you cannot shortchange in terms of going to market, and you need to do all the necessary activities and have a robust process and do full characterization. And what we have been able to do for some of our clients who have breakthrough status, for example, for their product, is be able to shave off the process characterization timeline by 3 months or 6 months even. And the way we have done that is leveraging the high throughput microbioreactor systems, AMBER systems that we use for process characterization. So there are other companies who have looked into this approach, but we are actually doing this for filing, and we have been very successful. So what that allows us to do is 24 to 48 reactors that you can run at a time, as opposed to 8, 12, or 16, and that allows much faster progress. And having the upstream high throughput married with analytical high throughput is really what has allowed us to move very fast and not again compromise on the quality side of things.
And can you speak further to the role that high throughput technologies play in reducing the product development timeline?
Absolutely. So as I was mentioning, the AMBER technology, the TCAN system that we use in downstream, as well as the octet-based system and TCAN for analytical capabilities, being able to not just have those equipment, but being able to really utilize them to their sort of maximum potential has been very crucial for us for both early and late phase sort of this expedited development approach. The other thing that we have been able to do is essentially become as hands-free as possible. So even if you have this equipment, in between you may need a lot of manual manipulation of samples from one room to another, etc. And what we are now going towards is moving completely hands-free if possible, so that the samples from AMBER goes directly to analytical TCAN and or goes to predictor plates for purification, then to TCAN for analysis, all hands-free, and that makes for a much more faster and more, frankly, higher precision, higher accuracy process.
Thank you.
Thank you.
Was there anything else you'd like to tell us?
Sure. So we have been able to go from transfection to GMP vial thaw in 9 months. We hope to even make that faster. And then in late phase, we are hoping to really create the benchmark for moving fast to BLA. So thanks for talking to me.
Okay. Well, thank you for talking with us this morning. and I hope you enjoy the rest of the show.
Thank you.